Okay, so I want to welcome everyone today. You're joining the second of our Urban Agriculture Pest Management webinar series. This is coordinated by the Great Lakes Urban Agriculture IPM Working Group, and this work is supported by the USDA National Institute of Food and Agriculture, specifically the Crop Protection and Pest Management Program through the North Central IPM Center, and we thank them for their support, of course. Just to give you all, since I've been letting new people in as we go here, just to give you all an orientation to our Zoom meeting today, we are asking for you just to stop your camera as you come on, so that we kind of have our webinar appearance, right? Because we are recording this to share out with folks who couldn't be here with us today. So we're just asking you to turn off your video. And also to mute yourself so that we don't have any unnecessary background noise. Okay. And once we get started here, I'm actually gonna lock that setting in place so that throughout the meeting, you won't be able to unmute yourself. So our primary form of communication with Michelle throughout the presentation is gonna be the chat box. If you have a question or need a clarification as we're going, I will be monitoring the chat box. So I will kind of unmute myself and ask Michelle your questions on behalf of you, okay? So with that brief orientation, let me tell you a little bit about our speaker today. Okay, so today, Michelle Wallace is going to talk about diagnosing nutrient deficiencies in plants. Michelle is the Northwest Ohio Ag and Natural Resource Educator with Central State University Extension. She works with beginning farmers, small farmers, specialty crops, alternative agriculture, and underserved populations. She has been working with CSU Extension since 2020. Before this, she worked for North Carolina State University for over 10 years. She grew up on a large, diversified farm overseas and spent a good bit of time working in the apple orchards and kiwi fruit vineyards with her father. So with that, I will turn it over to Michelle, and feel free to take it away and let me know if you need anything.
All right, can everyone see my screen? And everyone can hear me?
Looks great, and I can hear you. And if anyone, yep, I see some yeses in the chat box.
Okay, good. With the chat box in mind, I want to bring your attention. There is a handout that I put in the chat box. It is a visual, mobile versus immobile nutrient key. Towards the end of the talk or midway through the talk, I'm going to be asking everyone to pull up that key because we're going to use it with the interactive exercise. I usually do this talk in small groups. I've done it once at a large conference, but not. I haven't done it too much in the virtual world, so we'll see how it goes. But this talk isn't to replace the need for soil testing. It is just to help you figure out the problems that you sometimes see in the field with your plants and help give you the tools needed to at least reduce the number of possibilities to helping you solve what your problems might be. All right, so let's get started. So even though the focus of the talk is nutrient deficiencies, what I want you to know is it's really important to figure out if it's another problem. It could be a disease, it could be caused by insects, It could be abiotic related, and that means non-biological. So yes, nutrient deficiencies would fall in the abiotic realm, but it could be problems caused by other factors like weather. So there's a lot of different possibilities. And so when we go through this, I want you to think, like, pretend you're Sherlock Holmes and think through problem solving with me. And this process, this diagnostic process is very similar to what physicians go through when you go to, you have a medical visit. You have to identify the host. So when you're talking plants, you need to, um, narrowing down which plant the problem is occurring on helps you narrow down the problems, because you can look up to see what disease problems there might be, what insects problems this particular plant might have, describing the symptoms that you're seeing, reviewing what kind of history you have in your records. These things help you narrow down the problem. Don't assume anything at this stage, but by having all of the possibilities, it helps you focus in, because the problems are often limited by the host. So we want to be cautious, especially if you have a farm and you're seeing this at your farm, because if you jump to conclusions too quickly and make the decision that what you're seeing is because of an insect problem, and then you go and spray for that insect, or if you think it's a disease problem, and it turns out not to be, then you just spent time and money on a treatment that wasn't necessary. And that's also an environmental, issue. So you really want to gather all the data and information you can before jumping to conclusions that has economic and environmental impacts. So there's while there may be many pests that are that could cause problems to a particular plant, a lot of insect pests and disease pests are very host specific. Not all of them, but many of them have a particular palate. They only like to feed on specific crops. So by starting off identifying the host, you can narrow down a lot of the the possibilities as far as the pests are concerned. When you start to see multiple different hosts that are impacted, you need to start thinking about what other factors may be at play. Did your neighbor who has a large farm spray their farm with a pesticide that may have drifted over and settled on your crop? Did you clean your if you applied a pesticide, let's say a herbicide, to a weed, did you use a different container to spread it, or did you use the same one, and did you clean, is all the residue gone? So sometimes we inadvertently caused the problem that we're saying was too much fertilizer applied or not enough, or all of a sudden did it get cold and did that cause the problem? So we want to figure out what the source of the problem may be if we're not seeing any evidence of a biological organism, and if we're seeing multiple different species being impacted. So understanding signs versus symptoms is really important. So a sign is the actual biological um, entity is present on the plant, on the crop. So in this picture, we're seeing a bunch of aphids. That is a sign that the pest is present. With insects, it's a lot more easy. Even though some insects are really small, we can usually see them and their presence. Um, with diseases, it can be much more difficult because most diseases we need a microscope in order to see them. So because of that, we are thinking in terms of what symptoms. The symptoms are the the changes, the physical changes that we're seeing in the plant that is causing us to be concerned that we might have a problem. So what parts of the plant are affected? Are the roots, the crown, the stem, the foliage? Are both sides or just the upper side, lower side? So just looking, understanding signs versus symptoms because we want to narrow down the possibilities. So again, signs mean the organism is present. Symptoms are the visual effect of whatever is happening to the plant. It's what we can see, like, changing of the coloring of the plant, wilting, dieback, those kind of things. So just know that there's a lot of things that can be similar, can have similar symptoms. In this particular slide, we're seeing a mottled looking leaf. Those mottled symptoms are also on this tomato. When you become familiar with certain color, size, distribution of certain problems, you can recognize that this looks like a virus. If you see it enough, you may not know which kind of virus, but you might look at it and say, "Oh, okay, this could be a virus." So that is it's still a symptom. In order to identify the virus, we'd have to send it off to a plant, insect, and disease lab where they can culture it or look under a microscope and actually physically see it to say that that is a sign. So when we're looking at problems, we also want to look and see what the distribution of the problem is within the field, how, what particular plant parts are impacted. Is it the entire plant? Is it certain portions? Is an entire bed affected, or is it scattered? These are things that if you need help diagnosing the problem, someone who is a plant pathologist is going to ask you to help them figure out the problem. Having a magnifying glass or an inexpensive hand lens will help you magnify things. It helps you see things that might be really tiny. Insects or spider mites, they're very small. Thrips are very small. And a lot of times, in order to see some of these problems or pests, we need help. There are clip-on lenses to smartphones that you can purchase, all different kinds, that will help you magnify them on your phone so that you can also take pictures of them. And even if you can't identify them, you can send them in to a plant, insect, and disease lab for diagnosis, and that will help you take the photo. Having records and keeping really good records will help you figure out what the problem is. making sure that you log down dates, information about previous crops, how it was installed, irrigation, weather. All of these things will help you keep track of problems. And also, the records from year to year will help you be aware, oh, I need to look out. This problem was happening at this time of year. So record keeping is always a really good method of keeping track of not only problems, but from year to year. So an abiotic problem is a problem that could be caused by weather. It could be caused by soil fertility and nutrient deficiencies. It could be cultural, so it could be because of pesticides, wrong plant planted in the wrong place, poor care, maybe irrigation was needed and plants didn't get irrigation, um, or they got too much irrigation, improper care, maybe it was damaged from mechanical equipment. So there's a lot of reasons that cause abiotic plant problems. So this is the point where we're transitioning into what the talk is focused on. So now that we've kind of narrowed down the what it's not, we've decided that we don't see any excrement from insects. We don't see any signs that make us think that it's a disease. We think we might have a nutrient problem, but we're not sure. So we can go through these steps because you can use a visual, immobile versus mobile nutrient key to narrow down nutrient deficiencies. And the way it works is you have to understand that mobile nutrient symptoms appear on older leaves, while immobile nutrient symptoms appear on newer leaves. So the mobile nutrients are primarily the macronutrients. We've got nitrogen, phosphorus, potassium, chloride, magnesium, and molybdenum. I hope I pronounced that right. I'm not a chemist. Then we've got the immobile nutrients. Those are the ones that are going to be in the soil. They're not going to leave the soil because in huge amounts. These are nutrients that plants need in trace amounts and are usually readily available in the soil. Not always, but in soil they're usually found. So we've got sulfur, calcium, iron, zinc, manganese, boron, copper, and nickel. And these symptoms are going to be found on the newer leaves. So the first thing that you've got to do when you look at the nutrient, the mobile nutrient key, and I hope at this point in time, you can click in the chat box on the handout that I put in the chat box and pull this up on your computer if you can fit both the presentation and the key at the same time. So on one side of the key, you've got the mobile nutrient key. And the way a key works is you ask yourselves yes, no questions to help you move through the key. When you answer yes, and it's all based on looking at the visual symptoms. You follow the yes arrows down. If you answer the question no, then you follow the no answer. And likewise, the same works for the immobile nutrients, which are on the other side of the sheet. The first question that you have to ask is, are these symptoms showing up on the old leaves on the plant or on the new leaves? So we're going to go through a few series because the way the key works is there's only going to be one answer for each type of symptom that you could be seeing. This is the immobile nutrient key side. Again, these symptoms are going to show up on newer leaves. And we follow the yes arrows and the no arrows depending on the visual. We read the question and then we follow the arrow. So hopefully we're going to do an exercise and you're going to help me figure out in the chat box what you think you're saying. So let's start. And all of these examples are nutrient deficiencies that we see on tomatoes. A lot of people grow tomatoes. With different plants, the symptoms might look a little bit different. So some of your challenge will be in getting to know what they look like on the plants that you're growing. But tomatoes, a lot of people grow tomatoes, so I thought this would be a good exercise using tomatoes. So when we look at these tomato plants, the first thing we want to ask ourselves is, are we seeing the symptoms on old or young leaves? And you can see here, we're seeing what looks like to be maybe a spent flower here. So what do you think? Are we? I can't see. By the way, I can't see the chat from where I'm looking. So if Maggie, if you could tell me what people are answering in the chat, that would be great.
Absolutely. I got my eye on it.
So what do you think? Are we looking at old leaves or young leaves?
We've got, I don't want to bias anyone, but we've got a lot of old coming in.
Okay, so we're looking at old leaves. So that is correct. So because we're looking at old leaves, we're going to use the mobile key. And we're saying that the leaves are turning a little purplish on the underside, then on the top. The leaves may curl a little and the plants are growing really slowly. So let's look at that mobile nutrient key here. It can everyone, or those of you can look at your key. The first question we answered was older leaves are effective. We said yes, so we go down, and then it says the effects mostly generalized. Plants are dark or light green. What do you think? Are the plants dark or light green, or would you say the effects are localized chlorosis with or without spotting? Anyone? So we know, because I've said on the top here that it's a phosphorus deficiency. We say that the plants are dark green, often becoming purple and red. Does
Dark.
everyone see that and agree?
Yep, that's what's coming in the chat box. Dark.
Okay. All right. So now here we're looking at another plant nutrient deficiency. The plant, these are again tomato leaves. How would you describe the symptoms? Are they appearing on old or young leaves? If you could just look, you can look in the background. You see some flowers.
We've got young coming in across the
Okay,
board.
it's all over the place. So
Someone says both.
someone says,
Both. Most people are saying young. There was like 5 youngs. One person said both with a question mark. They weren't sure.
okay, so the challenge that we have here is that I have one picture, and sometimes it's easier to see if we see the whole plant. But these are actually on older leaves. Okay, and you can look and see here we've got signs of flowering. So we have older leaves, which would mean that you would use the mobile nutrient key. So we want to remember that nitrogen is very mobile. It goes from a gas in the atmosphere to a liquid on a dime. So every time it rains, nitrogen is converted from a gas to a liquid, and it rains nitrogen on our plants, and the roots of those plants take up that nitrogen. So when we see a nitrogen deficiency in a plant, what could be the causes of that? Is it, did it rain? Has it rained recently? We had last year a really, really long drought at least in Northwest Ohio. We went like, I don't know, 6 weeks without rain. So if you're fertilizing your plants, but you're depending on some of those nutrients like nitrogen to come from rain, that's when you might see nutrient deficiencies, like this blanching of the leaves is, it hasn't rained in a while, and we're starting to see problems. So you'll see the lower leaves turn yellow first, and gradually it will move upward.
Hey,
And this,
Michelle,
yes.
we had a great question that's kind of stepping back. And the question was about how do you know what leaves are young versus old?
So you're okay. So on plants, the the leaves that appear closer to the base of the plant are going to be old, and as the plant grows, the ones that are going to appear closer to the top of the plant are going to be the newer leaves.
Hmm.
Does that?
Yeah, for tomatoes and plants like that, I can't think of. I'm trying to think of other examples that might be a little different, like maybe like a cabbage, right? Those new leaves are put on in the middle, I think.
Oh, that is interesting. Um,
It's wherever that growth point is, but you can
right.
usually tell by observing the plant, right, as it grows. So,
Yes.
you know, where it's kind of putting on those new, really bright green leaves.
Yes.
Great question.
That's a great question. So when you key out this, you would look at the mobile nutrient key, and you would start by identifying that the older leaves are affected. Then you would follow the yes. Is it mostly dark green or light green? You would say um, light green. And so you would follow the yes line, and you would follow it down. Plants dark green often becoming purple or red, and you would say no. So you would follow the no arrow down. Plants are light green with leaves light green or yellow, no necrotic spotting. And we would say yes, and then we would find out that yes, this is nitrogen deficiency. So now we look at this leaf sample. And what are you seeing here? I don't have a picture of the whole plant. So it would be harder for you to key out that, is this an old leaf or a newer leaf? So I'm just going to tell you that this is an older leaf. But let's look at the symptoms that we're seeing. We are seeing inner venal chlorosis of the leaves. Does everyone see that? That's when we can see prominent veins, and in between the veins is the yellowing. Okay. Now we see this symptom a lot on young leaves too, but the difference is this sample is a sample of an older leaf. So if you had a whole plant that you were looking at, and you could tell that this is an old leaf, not a new leaf, that is where you start and decide which side of the visual key to use. So magnesium deficiency is often confused with iron deficiency. The big difference is magnesium um, is mobile while iron is immobile. So that's the big difference, and that's how you would know to use the immobile the mobile nutrient key. So we've got intervenal chlorosis. And this is particular problem is often caused when there is high levels of potash fertilizer, which locks up the nutrients. So these are sometimes over fertilization is the cause of our problems, or sometimes it's just the soil is very rich in nutrients, which are salts. And so that's sometimes what causes a nutrient deficiency. So here we've got another example of a nutrient deficiency. And what are we seeing? If we look at this plant, we're seeing the yellowing of the leaf on the inner along the midrib of the tomato plant, and then it's fading out towards the edges to dark green. So this is a young plant or an old plant. What do you think?
All right, I'm ready to see what's coming in. Ooh, we have some disagreements. Let's see. Mostly are saying young right now. One person said old.
All right, so we see some buds and for tomato flowering. So we are looking at younger leaves, and we're seeing the chlorosis going along the midrib. And then if you look at some of these pictures, you're looking at you can see the midribs and the yellowing around it. So you would, to solve this, you want to flip your key to the immobile nutrient key, which is used for the younger leaves. And you would go through, and you already answered that it's on younger leaves. And then you would look and you'd say, okay, is the terminal bud growing point, is it dead or alive? And we look at these buds here and they look alive. So we're going to say no, the growing point typically stays alive. Yes. Is chlorosis with or without intervenal chlorosis? So we've got here that we follow the no, because we want to follow the young leaves with intervenal chlorosis. And then we say yes. And then we see that we've got sharp distinction between the veins and the chlorotic areas. So I'm not sure that this example shows it as well as maybe it could, but this was the best example I could find of the intervenal chlorosis for iron. So hopefully you can see that. So here we've got another example, and I included the fruit because that is a lot of people may recognize this problem from the fruit. Can any of you recognize this nutrient deficiency just by looking at the fruit? Does anyone
Yeah, we've
have?
got calcium coming in. Yep.
Okay,
And
so
there's a blossom end rot. Yep.
blossom and rot. So a lot of people, when they see this problem, it's kind of too late to make any changes to impact these fruit, but you can a growing season is long, you can help mitigate this problem. So this is calcium deficiency, which is calcium is, what is it? Is it mobile or immobile? So calcium is immobile. It is, that means it's red. Usually we're seeing this plant problem where we have lots of calcium in the soil. So why do you think when we're seeing a calcium deficiency on a tomato with a nutrient that is immobile? That means it's usually available in the soil, readily existing, sometimes even at high levels. Why do you think we are seeing this problem when we know calcium is in the soil almost always? And does anyone have any thoughts on this before I tell you why?
Looks like we've got some blossom end rot experts in here. So a lot of talk of irrigation and uneven watering.
That's exactly right. So calcium is usually in our soils in large numbers, but when we see problems with the nutrient deficiency, it is because typically of irregular watering. It's not getting water in even amounts, and that water makes those nutrients available to the plant. So that is, you guys know your stuff. So when you're looking at a calcium deficiency, you're seeing blossom end rot, often caused by irregular calcium. You can correct this with a foliar spray, but know that there's typically plenty of calcium in the soil, and that the real issue is water. So checking your irrigation system, making sure things aren't clogged, making sure that water's getting to the plants is really important. On the immobile, you just, again, you go through the yes, no questions. One of the questions is looking, is there dieback of the buds? Yes. So just looking and going through and answering the yes, no questions on the key will help you key out these problems. So since we're talking about nutrient deficiencies, what I want to point out to you is most nutrients are available to plants when the pH is in the 6.2 to 6.7 range. In that pH range, most nutrients, both macro and micro, are available to the plant. When you go below 6.2 or above 7, so once you start going pH of your soils below 6 and above 7, you can start to see different deficiencies. You can start when it's below 6. You can see here that phosphorus is starting to become a problem. You might have phosphorus in your soil, but plants have a harder time getting it when the pH is low. Likewise, when we go over 7, we start to see problems with iron deficiency and manganese. So making sure when you get your soil tested that you're working towards adjusting your soil pH so that's in the ideal range is going to be one of your goals. Now, while some of you may have soil pHs below 6, most of my experience in Northwest Ohio is seeing soils with pH above 7. So to adjust those soil pHs, you would use elemental sulfur. For raising, that's for lowering the pH, and for raising the pH, it's you would use lime, but I haven't seen too much of that here in Ohio. But soil is different all over the place, and I always recommend that you soil test. When you soil test, you want to collect a random sample. So you want to choose, make sure that your sample is going to be for the crops that you're growing. So if you're growing the same crop, you want to get a sample of, if you have a small vegetable garden or a small farm and it's a diversity of vegetables and the soil is pretty uniform across the whole area, then you can sample all of the vegetables. But you want to go and collect samples of soil down between to the where the roots are in the ground, so down to about 6 to 12 inches below the soil surface. And you want to get samples from several different locations in the area where you are growing, so that you're trying to get a good sense of what's going on in the area. You want to put the soil in a plastic bucket, not a metal bucket, because the metal can cause trace amounts of metals to end up in your sample, and you don't want to impact the value of the readings that you're getting. You want the samples to not be overly wet when you mail them in. That also will make sure that it doesn't cost too much to mail them in. And you want them to be relatively dry for processing. So understanding a little bit about micronutrients can be helpful. There's one of the things that you'll see on your soil test report is this thing called CEC, which stands for cation exchange capacity. And it has to do with the nutrients in your soil are positively charged, and to help them be attracted, you want them um, to you want those nutrients to find a negatively charged clay particle, and so they have this exchange, so that the H nitrogen that happens when it rains or from water helps connect with these other cations, and this is what helps move up into the plant. So you've got different functions of the different nutrients have different functions for the plant. All of these functions are helpful with the plant in terms of photosynthesis, different reactions, different jobs within the plants. So, but this cation exchange capacity, these cations become available when water enters the soil solution. And you can see that certain soils have a higher cation exchange capacity. So soils that are high in organic matter have a lot more cation exchange capacity than the other soils. Clay has a really good cation exchange capacity, and sand is very poor in cation exchange capacity. So even though clay soils have problems with drainage that sometimes make farmers very upset, they do have a good amount of nutrients in them. Sometimes they just have a hard time draining water. So when you're looking at a soil test report, you want your cation exchange capacity to be over 10 and under 20. And we usually see that in soils that have clay and high organic matter. With that said, adding compost to all soils, unless they're really, really high already in organic matter, is really helps to improve the cation exchange capacity. When you diagnose a problem, or if you're seeing this problem and you're still unsure about using the key to confirm your results, what is recommended is to do a leaf tissue analysis, which involves collecting a fistful of leaves, placing them in a paper bag. Paper bag is actually kind of an important part because plastic bags can impact the quality of the sample. So you place it in a paper bag. Some plant problems, they want the petioles, some they don't. So these tests are usually done in the same labs as the soil testing and labs. So contacting them, asking, telling them what crop you're growing, and asking them whether or not they want the petioles, this end piece, included with the leaves or not, can be helpful. So in general, before you jump to using a key to diagnose a nutrient deficiency, make sure that your problem isn't caused by an organism like an insect or a disease. So make sure that you've narrowed down all the other possibilities. And once you've decided that you think you've got a nutrient deficiency, grab that key, keep it somewhere. I recommend, if you can, laminate it, but in general, keep it somewhere handy in a tool where you know where it is so you can grab it to help you narrow down the possible problems before you jump to a conclusion. And if you do find that it is a nutrient problem, before you go buy fertilizer, think to yourself, what are the reasons why you might be having a nutrient deficiency problem? Is it irrigation related? It also could be from too much fertilizer, especially in high tunnels where you start to see nutrient buildup at around the 5-year mark. That too much fertilizer can be the problem. And in that case, flooding the beds is also the answer. So I hope that gave you an idea. We have a little bit of time for questions if you want to ask them.
Absolutely. Yeah, let's open it up for questions. You can put your question in the chat box, or I can also allow you. I just changed it, so you should be able to unmute yourself. Yeah, but this is great, Michelle. I just had a question while you were talking about those micronutrients. It seems like it might be good to use that key, and then before almost you send your soil sample away, because you can request a lot of different things to be tested, right? So I'm sure there's a micronutrient.
That's right. So when you send your soil for sampling, they are going to test for macronutrients. They will never test for nitrogen, just so you know, because nitrogen, the plants take up what they need, and whatever they don't take up, it leaves the site pretty quickly. So they don't test for nitrogen. They just assume you're going to need it. But micronutrients are needed in trace amounts. I always recommend testing for both macro and micronutrients.
Great. And then there's some coming in, and I'll read these. Let's see. Oops, sorry, I'm trying to get to the first one that was asked. What's the best way to remove excess calcium, or is there?
Um, that's a great question. The only way you can slowly change the amount of calcium you have in your soil is by growing and planting crops. You're not going to really change it very much. You will start to see, especially if you use um, organic fertilizers made from um, animals, a buildup of metals like you'll see high calcium levels. You'll see, um, buildup of copper because what makes up the fertilizer is dependent on the food that they're eating. So, um, if you're using fertilizer made from chickens, for example, um, what they eat impacts what's what's in the fertilizer. So just thinking about these things will help you diversify, making sure that when you use fertilizer, if you have high calcium numbers or high any numbers, looking at what's in the fertilizer makeup before you put it out is helpful.
Mhm. Yeah, and Oregon State has a really great guide to natural organic fertilizer sources and how much, like you said, it's just an estimate because, each type of manure or each type of, um, naturally occurring fertilizer is going to differ, but they have a really great publication that estimates those things. So you can know if you're getting some horse manure free from down the road about how much of those nutrient values it might have.
Be wary of horse manure. I,
Right.
I've seen a lot of people have problems with um, herbicide residue transferring from composted horse manure
Okay.
into the production system.
Okay. Good. Good warning.
So if you get it, ask them lots of questions. Was what were the horses fed?
Someone just asked about recommendations for soil labs to use if they're sampling in Ohio.
So, so just so you know, Ohio doesn't have a government operated soil testing lab. Um, the states that surround us do have those. So you do have the option of sending it to Michigan, Pennsylvania, Indiana, and Kentucky. In Ohio, you have to send it to a privately operated soil testing lab. And I don't think I can give endorsements to any one in particular. I think I think Ohio State has. Do you guys have in your offices a list of different soil labs that they can send to?
Absolutely. We have a fact sheet, which I'll send out with the recording today, in addition to some of the other resources we're mentioning. But yeah, we have a soil testing fact sheet that has a list of um, Ohio and also the surrounding states certified labs, though. They're not just private. Some of them are private, some of them are university, but they're all uh, certified labs that can be trusted. Absolutely. Before anyone goes, I'm going to just launch this poll, um, just asking about, you know, if and what you learned today. I'm sure everyone learned something. But if you could just take that quick for yes, no questions, and we'll keep talking through the questions in the chat box as you guys do that as well. Um, so someone had a question about cation exchange, which is always a fun topic. Um, so what's the effect of organics? And I'm guessing like organic matter on cation exchange. Um,
So soils that are high in organic matter improve the cation exchange capacity. They make the 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 positive and negative charges are able to find each other faster, I guess is the best way to say. They happen more readily. But you have to understand that having once you go over the index number of 20, too much isn't always a good thing, and then you have an issue. So having organic, have lots of organic matter in the soil is great, and but it it works to when your index number goes over 20, then you start to see the tying up of nutrients, and so it's not as efficient, I guess is the best way I can describe it.
Mhm.
So no charges, no positive and negative charges in the soil is a problem. Like sand is not got much organic matter in it, and therefore there there's poor nutrients, there's usually poor growth. So we have to add compost organic matter to help improve the exchanging of cations. Clay has a lot of nutrients in it, but if your clay doesn't have a lot of organic matter, then clay also can benefit uh, by with the increase of organic matter, so that there's more exchange of cations. Those cations help the plants take up the nutrients.
So someone had a question, and I think this is a good one. How do you tell the difference between a disease and a nutrient deficiency? Like, how
So
can you look for that?
that's a great question. Okay, so and so to be clear, I'm not a plant pathologist, and but I will say that some of it involves um, how how are the symptoms distributed? Are they widespread across many plants, or are they localized and just in on a few plants? Is the pattern might also um, reflect, are we seeing a repeating pattern of symptoms, or is it localized to just a few plants? So a lot of it involves a little bit of pattern recognition, and some of it also, are we seeing multiple species of plants affected, or if it is it just one specific type of plant affected?
Mm.
Because abiotic symptoms tend to show up on a wide spread of plants, whereas diseases would be very host specific.
Absolutely. And sometimes you can actually see the signs of the disease, right? You can see some fungal um, threads or different parts of the reproductive disease. And have you ever done where you can put it in a plastic bag and see if it kind of grows a little bit more with like a paper towel?
So yeah, you can try to culture it yourself. Some disease problems, if you are on the roots, you might have to sacrifice a plant to see if you see the little nodules, like nematodes on the roots. Sometimes with bacterial diseases, you'll see a part of the stem. If you look closely at the plant and you're seeing kind of a part of the stem turning black, you could take a knife and cut it. You want to clean your knife afterwards. But if you see the center of the stem is turned black, then you know that you might have a problem with mushrooms. With sometimes you can see with fungal diseases, you can see these signs, not just the symptoms.
Mhm. Yeah, and also in some states there are plant pathology labs where you can send a sample. Um, and professionals then can culture and really give you a specific answer.
And Ohio State University has a plant insect disease lab at the Worcester campus.
Mhm.
It is for farmers free,
Mm.
but if you can take pictures first, I think they like to get there's and they want you to use your local extension educator to first help you figure out the problem before they get it. I
That's
think
right.
I think there is a fee for home gardeners.
Yeah, there is, but it's something reasonable, like $25. So, um, let's see. And we are at time, so if you have to jump off, we completely understand, but we might maybe take a couple more questions if that's okay, Michelle.
Yeah,
Let's see. We had another question about the calcium. High calcium is usually not too much of a problem in,
right.
is it?
Now I've seen soil test reports where the calcium levels are over 3,000. Just you have to just keep in mind that calcium is all these nutrients. So there's salts, they're vitamins. I think when we think about fertilizer, the TV ads they make us think that they're food, and really plants are the only living organism on the planet that makes their own food. And these nutrients are more like vitamins. We need them, but and to help us grow. Um, but in high levels, they're really like salt. And when you go to the beach, I like to compare it to going to the beach and swallowing salt water. It can get us dehydrated. So that's why we want to make sure that we don't over fertilize, because we don't want to put too much salt in the soil, because then it can cause reverse osmosis and water to go from the plant into the soil and cause the plants to die. The question was about, calcium levels getting too high. I guess it depends on how high is too high, but I've seen some pretty high soil test reports with calcium, and on farms that still had plants that are growing and looking all fine. So you just got to mitigate it as best you can, because
There's a couple questions. Oh, go ahead.
there's really not a lot you can do. So
There's a couple questions that are mentioning well water as their irrigation source and being concerned about the mineral levels in that well water. Do you have any recommendations on that?
So it is a good practice, is also for food safety, to get your water tested once a year if you can. And so if you're on a well, it is a really good idea. The same, I think the health department will test it for food safety reasons, but the soil testing labs that we mentioned before, I think also test water for nutrients. I feel like, and Maggie, you might have some thoughts on this as well. In my experience working with farmers, the pH is more of an issue with water than the calcium buildup in water. pH is a real issue with also nutrient availability. So sometimes, because the water is has a really high pH of like 8, that for many growers can be a problem.
Yeah,
And that's a complicated problem to solve. And I think working with um, an expert might help. And I'm probably not that expert to help you solve that issue.
absolutely. Yeah, I don't have anything else to add besides also, OSU also has a fact sheet on testing well water too, which I can include in the follow-up as well. Slightly different set of labs that might be available for testing water versus soil and plant material. Yeah, but I think that was the last question. Everyone is putting great comments in the chat box of saying thank you, great presentation. A lot of people really appreciated your presentation and they were able to absorb this complicated information as they were able to go. So yeah, and I will send out the handout that Michelle shared with everyone, in addition to some of those resources we mentioned. So, Michelle, thank you so much. Is there anything you want to, anything else you want to add before we can?
I can't think of anything else, but thank you so much for letting me give this presentation. And I hope that you all use the key and help you think about, really think about the problems and understand that there are many possibilities and there are very scientific ways to go about eliminating some of the possibilities. It just takes practice.
Hmm. Absolutely. Well, I just want to give a reminder that we have one more presentation in our series that's next week. And that presentation is going to be with Dr. Erin Hill from Michigan State University. She's going to talk to us about apps that identify plants and weeds, especially. So I know that'll be a really interesting one to join as well. Um, so we hope to see you next week. And Susan, you're just asking about where to get the key. I'm going to send that out via email. So I will stick to what I said in my email and send this recording out by the end of today. And I'll send that with all of these resources as well. All right. All right, everyone. Thank you so much. And we hope to see you next week. Thanks again, Michelle.
Thanks. Bye.